Today, first of all, I would like to repeat a little bit what we did last time. So, and also finish the topic which is related with action potential, transduction of action potential, and so on. I will also ask you to answer some questions. So it's a chance to gain some points. The questions will be, of course, based on the information you received last lectures. And also, the second part of this lecture will be proteosynthesis. I already prepared the video because I am not sure about the stability of the Wi-Fi signal because I'm now teaching from home. And after that, also you will have, or you will receive by email the little questionnaire, as you probably know from previous time. Maybe I'm going to send it now if I have the internet. So now every one of you received the email with the video to proteosynthesis, but I will also show you the video also here in this lecture. And we will finish maybe 10 minutes earlier, and you will have time to go through the questionnaire. So questions? Okay, if not, we can start. Okay.
Excuse me, I would like to ask you about the exam.
About, maybe, could you repeat it, please? I didn't hear it.
Yeah, I would like to ask you about the exam, when we will have it.
Exam. Probably there will be no exam because you are, maybe I can show you that you are working continuously and you are gaining lots of information. Yeah, I didn't have time to check these calculations, but probably it will be okay. So today I will check it. And as you can see, for example, Marek, he has almost 30 points. So he needs 5 points, 5.5 to gain the credits. So probably there will be no test. Only I think Omar probably will write a test because he's not attending this lecture. He's not do anything. So I saw him once.
Sorry, actually, Omar, he will not attend anymore.
Okay, that's good information. Thank you very much.
You're welcome.
Okay, so I think probably the rest of you, I don't see any problem with your attendance, with your effort. Probably you will gain the credits or the important points until the end of this year or maybe in the beginning of the next year. So don't worry. So, Mariam, don't worry. So let's start today.
Thank you very much.
You are welcome. Okay. I think we have finished last time in, I think, okay, the last information was about mediated channels opening. So because they use some mediator like, so the signal is coming from adrenaline, noradrenaline, serotonin, so very powerful chemicals which are recognized as hormones. However, these hormones are not binded directly to the receptor which is present on the channel, but they are binding. They induce or indicate the cascade which induce G protein to stimulate and bind with the receptor present in the channel. So this mediated opening channels, they work slowly because there is this two-step regulation, and therefore some corrections of their opening or just keeping them closed might be provided. In general, they have a slower response. So I think that was the last question. Now I will ask you, maybe Marek, tell me how you can characterize the voltage-gated channels.
Like how they work or how they are structured?
Maybe how they work, what they control, how they are controlled, maybe just simple answer.
Well, it's the result of the potential difference of outside the cell and inside the cell.
Mm-hmm. Yep.
And so the movement happens thanks to this difference, which is, if I remember correctly, about 50 millivolts, minus 50 millivolts.
Yes, approximately like this. Yes, thank you, Mark. It's great. What about Mariam? Could you characterize, please, the operation or control of chemical control channels? What control the chemical channels or chemical operated channels?
So the chemical channels are determined by the charge.
Mm-hmm.
That it's
But it's not chemical control. It's not the charge. It's not the charge. Okay, maybe Eva can tell me the answer. What is the key point of the operation of the chemically controlled channels? What operate them? What make them open?
Well, some ligands or some chemical substances.
Mm-hmm.
So they're
Yeah, it's
membrane
fine. It's enough.
receptor.
Receive a point. Xiaomin, could you tell me 2 examples of okay, Xiaomin, tell me what is how we call in general the chemicals which control the activity of chemically controlled channels. Just one word. What is the general name for these chemicals? Mm-hmm. Okay. What about Katerina? Do you know the name for the chemicals which make the chemical control channels open? In general,
It's
Cadmium.
acetylchlorine.
it's just an example, okay, acetylcholine, but okay, do you know another like acetylcholine and maybe one another?
Glutamate.
Yes. So okay, and maybe I heard CMAN, but I didn't hear your answer. What is the general name for acetylcholine, GABA, glutamate, and so on? Okay. What about Mariam? The same question.
Could you please repeat it again, because the internet connection is very bad here?
It's very bad. Okay, maybe my last question today to you. You can answer. Who knows the answer? Just tell me. What is the general name for acetylcholine, GABA, and also glutamate, for example? General name for this group of chemicals.
Synaptic potentials?
No, it's neurotransmitters, guys. So I'm going to move a little bit because I have to find some better Wi-Fi signal. It will be a little bit better. So I hope. Is it a little bit better here? Guys, please give me a feedback if you can hear me and listen.
It's okay, yes.
Yes,
It's
we
better.
Yes, we can hear you.
Thank you. That's great. So let's finish this little talk, this little presentation about the channels. First of all, we have to mention also what happened with neurotransmitters. Now we are going to talk about the chemically controlled channels. Therefore, I ask you if you know how they operate, how they are operated or controlled. So they are opened by the binding of a neurotransmitter to the active site of the ion channel. So, for example, in acetylcholine channel or acetylcholine receptor, it's the 5 proteins unit. 2 subunits, which are called alpha, they have an active site, which is sensitive for the binding of acetylcholine. However, what happens if the acetylcholine is binded to the active site and the channel is open? So the sodium goes inside the cell, or and the membrane is depolarized. We explained it last time deeply. So now what happens if the channel has to be inactivated, and also the system has to go back to the negative potential, has to be repolarized again. So if acetylcholine stay there in the presynaptic cleft, the channel stay inactivated. So this is not really the physiological state. Therefore, we have an enzyme which is called esterase, acetylcholine esterase, which decompose acetylcholine to acetyl and choline, and it is then the channels are then reactivated. Of course, we have some chemicals which may block this enzyme, acetylcholine esterase, for example, sarin. This chemical which were firstly used as a chemical weapon in First World War, and this chemical blocks the acetylcholine esterase, so therefore acetylcholine cannot be decomposed, and the channel is blocked, and therefore the action and the working ability of whole synapses is blocked. Similar effect has also some insecticide, for example, Diffuzil. Probably you know it's a trademark. It's the Czech chemicals which is developed as a protection against ticks and some mosquitoes. It works for these little animals. Yeah, it's a version for those animals. The second option is how to reactivate the synapse is the resorption back to synapses. This is another way how to do it. However, this strategy is not applied for acetylcholine. This is applied for example, for glycine. What about curare? Probably you heard about some chemicals like poisons, which may also paralyze the whole body. Curare it's the nature product from frogs, which is widely used in South American Indians. They use it for the paralysis of animals. They shoot them with arrows, and the arrow doesn't kill the animal. However, the animal is paralyzed by curare. Curare works not the same way as sarin. Curare has the binding activity the same as the acetylcholine. So it means it blocks the receptor for acetylcholine. So the way how curare works, it's not just the inactivation of the channels, but the blockage of them, and they are kept closed. The second chemical which is well known, it's strychnine. Probably if you read some books from Agatha Christie, you heard that there are some chemicals which can be used as a poison. It's strychnine. This chemical also blocks by the same way as curare also the receptor for in this way glycine, not acetylcholine but glycine. So the result of the blockage of the glycine receptor by strychnine is the uncontrolled convulsions. So because the signal is spread via the synapse, because the glycine receptor is it's the suppressoric synapses. Another suppressoric synapses is GABA receptors. This is another group of channels. You heard about them last time. These channels are usually used for the transport or diffusion of chlorine ion. And we have some chemicals, usually artificially prepared barbiturates, rohypnol, something like that, which increase the efficiency of GABA, so gamma aminobutyric acid, and therefore the suppressoric synapses are activated. What is the result? If we just increase the efficiency of suppressoric synapses by barbiturates, for example, we just calm down, we just go asleep, and we are just I'm really a little bit afraid to say relax, but we just calm down because of the suppression of our activity. Now, this is what I already mentioned. So voltage control channels, Marek replied correctly. that they are open by the change of the voltage. It's just little change, around 15 millivolts, so from -70 to -55, something around, and this is enough just to open the sodium voltage gated channel, and therefore the development of the action potential is allowed in the first step. This is just the visualization how the sodium channel, which is voltage control, is open. So here you can see that this is the step number 1. I'm not typing. Give me a second. Okay. So here is the resting membrane state. The channel is closed, so sodium cannot go through. And then the potential is slightly changed, so the difference is something like 15 millivolts towards to the more positive potential, and the channel became open. So sodium goes through the potential and the membrane get to more and more positive potentials. So if the potential exceed the value which is in a positive range, something around 30 millivolts, plus 30 millivolts, the channel became inactivated. So it means that it is open, it is closed, but by different way that in the first step, that it is open by the inner conformation changes. I think it's visible, it's clearly visible from this part, because here you can see that it's a negative. Maybe I will put there negative. This is also negative, but a little bit towards to the more positive, and this is a positive potentials. So now I would like to just a little bit summarize what is the action potential. So first reaction comes to the voltage open potentials or voltage gated potentials, as you remember from previous, and also the high concentration of these channels is usually on the signal receiving parts, so the entering part, and also in the root of axon. So this is the initiation segment. Do you remember the picture of neuron? So we have the dendrites, we have the body of neuron, and there is the initiation segment. We can call it also like a neck of axon. This is the initiation segment where the concentration of these voltage-gated channels is pretty high, and therefore the convolution of the membrane potential, of the membrane action potential, can be provided. So this is just another picture as I showed you previously. So what happened? Firstly, the sodium channels are open. Then more sodium channels are open because it's the positive feedback. Once one is open, so it's like an explosive reaction. Then the sodium channels it's closed, but it's better to say that they are inactivated. That's also the signal for the potassium channels, but the signal is not coming from the closure of sodium channels, but it's coming from the value of the potential. So the potassium channels became open. Again, potassium is going through the membrane, and finally, in some level of potential, the potassium channels are inactivated. Then we have to start the recovery of the system, and this is the way how to apply the sodium-potassium pump, because it's pumping sodium outside the cell and potassium inside the cell. And this process is finished by the hyperpolarization of the membrane. And however, it's pretty short time when the membrane is hyperpolarized, and in a very short time, in milliseconds, it returns to the normal membrane state. Also, we have to talk about the propagation of the action potential. I mentioned last time that we have two types of axons. the older one or evolutionally older one, which is in sepia, for example, and lower animals, there is no myelin sheath, and the action potential is propagated along whole the axon to the end of the axon to the synapses, for example. The speed of this propagation is pretty high, and it's increasing with the diameter of this axon. However, for higher animals like humans, elephants, and so on, this speed is not enough because you have to find some compromise between the diameter of axons and also the rate of the propagation of the speed. That's the reason that in higher animals we can observe some insulation, particle insulation of axon, and therefore the signal just jump from one polar place to another one, just to overjump the insulated part, which is called the myelin sheath. The myelin sheath is created by Schwann cells, and only the part which can be polarized is the Ranvier knots. Like that. So here you have the myelin sheath. This is an insulator, so it's this substance is rich to fats, so therefore no electric charge can be transferred, no polarization can be occurred. But this part, the nodus of Ranvier, it's very irritative, very sensitive part, which is rich of sodium and potassium channels, and the development of action potential is provided exactly here. Sometimes it's quite difficult for people just to imagine how the charge can jump, but just to imagine that the distances between the knots are pretty short. So therefore, the jumping of the potential it's possible. So that's all. Now we are going to switch to a slightly different topic. Okay, and nerve terminal, it's a slide which will be used for the lecture, which is related with muscle and muscle contraction. So today I am going to switch to the video. So questions? Could you hear me, or I'm still online or not?
Yeah.
Yes.
Okay.
No questions.
So you have no questions? Okay. I think because the video I already send you the link, so it's not necessary to record it. I'm going to stop it. Okay.